Class 11, Chapter 8, the unit of life, cell. So, today we look the introduction. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what it is that makes an organism living? Or what is that inanimate thing does not which a living thing has? The answer to this presence of the basic unit of life, the basic unit of life, the cell in all living organisms. All living organisms are composed of cells. Some of the composed of single cell are called unicellular, whereas like others are composed of multicellular cells. Now, what is a cell? Unicellular organisms are capable of independent existence and performing essential function. Anything less than a complete structure of cell does not ensure independent living. Hence, cell is the fundamental structure and functional unit of living being. Antoine Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structures detail of cell. Then the cell theory. In 1838, Matthew Scalden and German botanist and examined a large number of plants, observed that all plants are composed of different kind of cells, form of tissues of plant at about the same time. Theodore Squadron, a British zoologist, studied different types of animal cell and reported that cell had a thin outer layer, which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that presence of cell wall is unique character in plant cells. On the basis of this, Squadron proposed the hypothesis of bodies of animal and plant composed of cells and product of cells. Scalden and Schwann together formulated cell theory. The theory, however, did not explain how new cells are formed. Rudolf Virchow 1855 first explained cell divide and new cells are formed from pre-existing cells. Omnis cellula e cellula. He modified the hypothesis of Scalden and Schwann to give the theory of final shape. The cell theory as understood today. All living organisms are composed of cells. 8.3 An overview of cell. You have earlier observed cell is an onion peel of or human cheek cell under microscope. Let us recollect their structure. The onion cell, which is typically plant cell, has distinct cell wall as outer boundary and is within its cell membrane. The cell of human cheek have outer membrane as delimiting structure of cell. Inside each cell is dense membrane bound structure called nucleus. The nucleus contains chromosomes which turns into genetic material. DNA cells that have membrane-bounded nuclei are called eukaryotic, whereas cells that lack of membrane-bound nucleolus are prokaryotic. In both prokaryotic and eukaryotic, semi-fluid matrix called cytoplasm occupies the volume of cells. Cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occurs in keeping of the cell in the living state. Besides the nucleus, the eukaryotic cells have outer membrane distinct structure called organelles, like the endoplasmic reticulum, Golgi apparatus, Golgi complex, lysosome, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all both eukaryotic as well as prokaryotic. Within the cell, ribosomes are not found only in cytoplasm but also within two organelles, chloroplast and mitochondria, or on rough endoplasmic reticulum. Animal cells contains another non-membrane bound organelle is called centriosome, which helps in cell division. Cells differ greatly in size, shape, activities. For example, mycoplasm, the smallest cell is 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is egg of ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cell is the longest cell. Cells vary greatly in shape and size. They may be disc-like, polygonal, columnar, cuboidal, thread-like, or even irregular. The shape of cell may vary to the function they perform. And here is diagram 8.1 showing different shape of cell. Nerve cell, branched and long, red blood cell, round and concave, biconcave. White blood cells, amoeboid. Columnar epithelial cells, long and narrow, are tracheaed, elongated. Mesophyll cells, round. 8.5.5 Plastids. Plastids are found in all plant cells and euglenoids. These are easily observed under microscope as they are large. They bear some specific pigment, thus imparting specific colors on plant. Based on the type of pigment, plastids can be classified into chloroplast, chromoplast, leucoplast. The chloroplast contains chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoids pigments like carotenoids, xanthophylls and other are present. The gives part of plant yellow, orange, red color. The leucoplast is colorless plasters of varied shape and size stored in nutrition. Ameloplast store carbohydrates, starch, example potato. Usually plast stores oils and fats, whereas the alliloplast stores protein. Majorly, all of the chloroplasts are green plant are found in mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid and even ribbon-like organelles have variable length 5 to 10 micrometer and width 2 to 4 micrometer. The number varies from 1 per cell of the Chlamydomonas, a green algae, to 20 to 40 per cell in the mesophyll. Like mitochondria, chloroplasts are also double membrane bound. Of the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in stroma. Thylakoids are arranged in stacks like piles of coins called grana or integrated thylakoids. In addition, there are flat membrane tubules called stroma lamellae connecting the thylakoids of different grana. The membrane of thylakoids enclosed space are called lumen. The stroma of the chloroplast contain enzymes required for the synthesis of carbohydrates and protein. It also contains small double-stranded circular DNA and ribosomes. Chloroplast pigments are present in thylakoid. Ribosomes of chloroplast are smaller, 70s, than cytoplasm ribosome. Cytoplasmic ribosome is 80s. Ribosomes. 8.5.6 Ribosomes are granular structures first observed under electron microscope as dense particles by George Palade. They are in 1953. They are composed of ribonucleic acid, RNA and proteins and are surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosome are 70s. Each ribosome has two subunits, larger and smaller subunits. The two subunits of 80s ribosome are 60s and 40s, and 70s are 80s and 30s. The S is Svedberg's unit, stands for sedimentation coefficient. It is indirectly measured of density and size. Both 70s and 80s are composed of two subunits. 8.5.7 Cytoskeletal. An elaborate structure of filament proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of cell. Figure 8.10 says section of cilia flagella showing parts, electron micrograph, diagrammatic representation of integral structures. 8.5.8 Cilia and flagellae. Cilia and flagellae are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either of the cell surrounding fluid. Flagellae are comparatively longer and are responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that eukaryotic flagella. The electron microscope study of cilium or the flagellum show that they are covered with plasma membrane. Their core, called axoneme, possess a number of microtubules running in parallel long axis. The axonema usually has 9 doublets radially arranged peripherally microtubules and a pair of eukaryotic cells. The eukaryotics include the protista, plantae, animal and fungi. The eukaryotic cells, there are extensive compartmentalization of cytoplasm. Through the presence of membrane-bound organelles, eukaryotic cells possess of an organized nucleus with nuclear envelope. In addition to eukaryotic cell, have variety of complex locomotory and cytoskeletal structures. The genetic material are organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as form of possess of cell walls, plastids and large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent almost in all plants. Then figure 8.3. A plant cell and B animal cell. What are the components of plant cell? Rough endoplasmic reticulum, smooth endoplasmic reticulum, lysosome, plasmodesmata, microtubule, perioxosome, cytoplasm, chloroplast, ribosomes, mitochondria, cell ball, middle lamella, vacuole, plasma membrane, nuclear envelope, Golgi apparatus, nucleolus and nucleus. Then is animal cell. It have Golgi apparatus, a smooth endoplasmic reticulum, nuclear envelope, nucleolus, nucleus, cytoplasm, rough endoplasmic reticulum, mitochondria, ribosome, lysosome, perioxosome, centriole, plasma membrane, microvilli. Then cell membrane. The detailed structure of membrane was studied only after the advent of the electron microscope in the 1950. Meanwhile, chemical studies on the cell membrane, especially in human red blood cell, enabled the scientists to deduct the possible structure of plasma membrane. These studies showed that cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in bilayer. Also, lipids are arranged within the membrane with the polar head towards outer side and the hydrophobia tails towards the inner side. This ensures that non-polar tail of saturated hydrocarbon is protected from aqueous environment. In addition to phospholipid, membrane also contains cholesterol. Later biochemical investigation clears that reveals cell membrane also possess protein and carbohydrate. The ratio of protein lipid considered in different cells. In human being, the membrane of erythrocyte has approximately 52% protein and 40% lipid depending upon ease of extraction. Membrane protein can be classified as integral and peripheral. Peripheral protein lie in the surface of membrane, while the integral protein are partially or fully buried into the membrane. Then, an improved model of structure of cell membrane was proposed by Singer and Nicholson in 1972. Widely accepted as fluid mosaic model. According to these, the quasi-fluid nature of lipid enabled lateral movement of protein within the overall bilayer. The ability to move within the membrane is measured as fluidity. The fluid nature of the membrane is also important for the point of view of junction like cell growth, formation of intercellular junction, secretion, endocytosis, cell division. One of the most important functions of plasma membrane is to transport of molecule across it. The membrane is selectively permeable to some of the molecule present on either side of it. Many molecules can be brief across the membrane without any requirement of cell, and this is called passive transport. Neutral solutes may across the membrane by the process of simple diffusion along concentration gradient from higher concentration to lower. Water may also move across the membrane from higher concentration to lower concentration. Movement of water by diffusion is called osmosis. As polar molecule cannot pass through the non-polar bilipid layer, the required carrier protein of the membrane facilitates the transport across the membrane of few ions or molecule are transported across the membrane. Again, the concentration gradient from lower to higher concentration such a transport energy dependent process in which ATP utilized is called active transportation. Example: sodium potassium pump. Cell wall. As you may recall, a non-living rigid structure called cell wall formed an outer layer covering from the plasma membrane of fungi and plants. Cell wall not only given shape to cell and protect cell from mechanic damage and infection, it also help in cell-to-cell interaction and provides barrier undesirable macromolecules. Algae have cell wall made of cellulose, lactase, mannose, and minerals like calcium carbonate, while other plant consist cellulose, hemicellulose, pectins, proteins. The cell wall of young plant is primary wall, the capable of growth which gradually diminished as cell mature, and the secondary wall formed on inner side of the cell. The middle lamella layer of calcium pectate which holds glues of different neighboring together. The cell wall of middle lamella may be transversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While the membranous organelles is distinct in terms of its structure and functions, many of these are considered together as endomembrane system because their function and coordination. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Since the function of mitochondria, chloroplast, and peroxisome are not coordinated with the above components, these are not considered as part of endomembrane system. Endoplasmic reticulum. Electron microscope studied in eukaryotic cells reveal the presence of network of reticulum of tiny tubular structures scattered in cytoplasm that is called endoplasmic reticulum. Hence, ER divides intracellular space into two distinct compartments, luminal and extraluminal. Luminal is inside endoplasmic reticulum and extraluminal is cytoplasm compartment. The endoplasmic reticulum often shows ribosome attached to their outer surface. The endoplasmic reticulum bearing ribosome on the surface is called rough endoplasmic reticulum, and the absence ribosome they appear as smooth and they are smooth endoplasmic reticulum. RER is frequently observed in cells actively involved in protein synthesis and secretion. They are extensive, continuous, and outer membrane nucleus. The smooth endoplasmic reticulum is major site of synthesis of lipid in cell. Lipid-like steroidal hormone synthesized in SCR. Golgi apparatus. Camillo Golgi, 1898, first observed densely strained reticulum structure near nucleus. These were later named as Golgi body after him. They consist of many flat disc-like sacs or cisternae of 0.5 nanometer to 1 nanometer in diameter. These are stacked parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distant convex cysts of forming face and concave trans or maturing face. The cis and trans face of organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to intracellular targets or secretes outer the cell. Material to be packed in form of vesicle from ER fused with the cis face of Golgi apparatus. The movement of maturing phase, this explained by Golgi apparatus, remains in the close association with the endoplasmic reticulum. A number of proteins synthesized by ribosome on endoplasmic reticulum are modified by cisternae of Golgi apparatus before the release from transphase. Golgi apparatus is the important site of formation of glycoprotein and glycolipids. Lysosome These are membrane-bound vesicle structure formed in the process of packaging of Golgi apparatus. The isolated lysosomal vesicles have been found in very rich in amount of all these hydrolytic enzymes. Hydrolyses are lipase, protease, carbohydrate optimally active at all pH. The enzymes are capable of digesting carbohydrate, protein, lipids, and nucleic acid. Vacuoles. A vacuole is the membrane-bound space found in cytoplasm. It contains water sap, secretory products, and other material not useful for the cell. The vacuole is bound by single membrane called tonoplast. In plant cell, vacuole are occupy 90% of volume of cell. In plant, tonoplast facilitate transportation of number of ions in other material against the concentration gradient into vacuole. Hence, their concentration significantly higher in vacuole than the cytoplasm and ammonia. The contractile vacuole is important osmoregulatory and secretory in many cells, as it protista food vacuole are formed engulfing the food particles. Mitochondria. Mitochondria, unless specially stained, are not easily visible under microscope. The number of mitochondria per cell is variable depending on physiological activities of the cell. In terms of shape and size, considered degree of variation is observed. Typically, it is sausage shaped or cylindrical, have diameter 0.2 to 1 micrometer and length 1 to 4 micrometer. Each mitochondria is double membrane bound structure with the outer membrane and inner membrane divide its luminary distantly into two aqueous compartments, the outer compartment, inner compartment. The inner compartment filled with dense homologous substance called the matrix. Outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfolding called cistrae towards the matrix. The cistrae increase surface area. Two membranes have their own specific enzyme associated with mitochondrial function. Mitochondria are site of aerobic respiration. They produce cellular energy to form ATP. Hence, they are called powerhouse of cell. The matrix of process single circular DNA, few RNA, ribosome 70S are the component required to synthesize of protein. The mitochondria. Ribosomes and inclusion body. In prokaryotic, ribosomes are associated with plasma membrane of the cell. They are about 15 nanometer to 20 nanometer in size and are made of two subunits, 50S and 30S units, which present together by 70S prokaryotic ribosome. Ribosomes are site of protein synthesis. Several ribosomes may attach to single mRNA to form chain called polyribosome. The ribosome of polysome translate the mRNA into protein. Inclusion body. Hydrogen material in prokaryotic cells are stored in the cytoplasm in the form of inclusion body. These are not bound by any membrane system and live free in the cytoplasm. Example: phosphate granule, cyanophysium granule, and glycogen granule. Gas vacuole are found in blue, green, and purple and green photosynthetic bacteria. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO. Pneumonic-like organisms. They are generally smaller and multiply and rapidly than eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus spherical, vibrio comma-shaped, and spirillum spiral-shaped. The organism of prokaryotic cells is fundamentally similar, even though prokaryotic exhibit a wide variety of shapes and functions. All prokaryotic have cell wall surrounding the cell membrane except in the mycoplasma. The fluid matrix building in the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically not enveloped by a nuclear membrane. In addition to genome, DNA, single chromosome, circular DNA. Many bacteria have small circular DNA outside the genome DNA. The smaller DNA are called plasmids. The plasmid DNA confirms certain unique phenotypic characters to such bacteria. One such character is resident to antibiotic. In higher classes, you will learn about the plasmid DNA is used to monitor bacterial transformation. With foreign DNA, nuclear membrane is found in eukaryotics. No organelle-like ones in eukaryotic are found in prokaryotic except for ribosome. Prokaryotic have some unique in the form of inclusion, a specialized differentiate form of cell membrane called mesosome. The characteristics of periodics. These are essential enfoldings of cell membrane. Next is cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of tightly bound three-layered structure, the outermost glycocalyx, cell wall, plasma membrane. Although each layer is envelope formed of distinctive functions, they act together as simple protective unit. Bacteria can be classified into two groups on the basis of difference in cell envelope and the manner in which respond to staining procedure developed by Gram-Wis. Those that take up the Gram stain are Gram-positive and the others do not are Gram-negative bacteria. Glycocalyx differs in composition thickness among the different bacteria. It could be loose sheet called the slime layer, while in other could be tight and thick cell capsule. The cell wall determines shape and cells also provide a strong structural support to prevent bacterial from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. The membrane is similar structurally to that of the eukaryotics. A special membranous structure is the mesosome, which is formed by extension of plasma membrane into the cell. The extensions are in the form of vesicles, tubulin, lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion process to increase the surface area of plasma membrane and enzymatic content. In some prokaryotic like cyanobacteria, there are other membranous extension into the cytoplasm called chromatophore and contain pigment. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension, the cell called flagellae. Bacteria show a range of number of arrangement of flagellae. Bacterial flagellum is composed of three parts: filament, hook, basal body. The filament is the longest portion extended from the cell surface to the outside. Besides flagellae, pili, fimbriae are also structures of the bacteria that do not play in motility. The pili are agglutinated tubular structures made up of a special protein. The fimbriae is a small basal-like fiber sprouting of the cell. In some bacteria, they know help to attract bacteria to rock in steams and also the